So the chapter that we're going to look at is by David Myers, his 8th edition textbook, but the 9th and the 7th will go along just as well. So we're going to look at sensation, chapter 5. So remember, these are just outlines of what the chapter is and things that you should definitely know. So when we look at sensation and perception, sensation are things that we get, stimuluses that we get from our senses. So taste, hearing, touch, seeing, these are our sensations, and then how we perceive it is perception. So if I touch my cold water bottle, okay, the cold water bottle, the stimulus, okay, is going to now be percepted. I'm going to interpret how I touch that cold bottle. So the cold bottle is the sensation, you know, it's cold, I got the goosebumps going up my arms, and then my brain or other things help me interpret that that is a cold water bottle. So sensation and perception work together. So chapters 5 and 6 are a good portion of your AP exam, so make certain that you do go over these areas. There's a lot of neurobiology, there's a lot of biology components within this unit. So when we look at bottom-up processing, what this means is you analyze the stimulus, and it begins with the sense receptor. So I touch the cold water bottle, and it works up to the level of the brain and the mind. So cold water bottle, and then like what I'm touching it, now I'm interpreting it as a cold object. So top-down processing, what this is let's use the exact same example as the cold water bottle. What you do here is it begins with your preconceived perception, so with your perceptions, and then it works down. So I touch the water bottle, I know it's cold, and so now my sensation is cold. In this picture by Bev Doolittle, The Forest Has Eyes, you can see that what we're looking at here is a lot of different faces within this actual painting, and you guys picked out all these different faces within this picture. And so what sensation and perception working together does is it helps us sort out these complex images. So we're able to see, we're able to have a stimulus, and then our mind is able to perceive those stimuluses. So senses are our nature's gift to help us sort out and survive. So people are actually attracted to other people's odors. Everybody has a personal smell. And so a male is going to smell a female. And if that scent is what he likes, then that's how he is going to survive. Because remember, males will pass down their DNA, and females are the same way. Females are attracted to scent too. So when a female smells a certain scent that she likes, she is going to get with that man and pass her genes down as well. So exploring the senses, what stimuli cross our threshold for conscious awareness? Could we be influenced by stimuli too weak, subliminal to be perceived? And why are we unaware of unchanging stimuli like a band-aid on our skin? So psychophysics, so this is looking at the physical world and the psychological world. And so in the physical world, we got light, and in the psychological world, we call it brightness. So this is just the yin-yang, you know, the physical versus the psychological world. When we look here, we're going to look at detection, and we're going to look at intensity. So tell when you, the observer, detects the light. Okay. Most people perceive the light right here, okay? And that's the stimulus that you perceive. That's your absolute threshold. So absolute threshold is the minimum stimulus, or the minimum stimulation needed to detect a particular stimulus 50% of the time. So everyone in here has had a hearing test. And when you have a hearing test, it does that beep. The lowest beep that you can hear, that's your absolute threshold. The minimum amount of stimulation that it needs for you to actually detect something 50% of the time. Subliminal thresholds is when it's below your actual conscious awareness. So the people who do like taste testing for like Ben & Jerry or the Jelly Belly people or perfume smellers, those people are people who can actually get below one's absolute threshold for conscious awareness. So when stimuli are below one's absolute threshold for conscious awareness, you wouldn't even be able to taste that subtle cinnamon or whatever it is in the Jelly Belly or the Ben & Jerry's, whatever it is, or smell it. So the difference threshold, this is the difference that you can detect between two stimuli about 50% of the time. We also know the difference threshold is the just noticeable difference. So between this one and this one, this is what we call the difference threshold. So Weber's law, two stimuli must differ by a constant minimum percentage to be perceived as different. So the percent's got to be constant in order for people to actually see a difference. So signal detection theory predicts how and when we detect the presence of a faint stimuli, the signal, amid background noises, other stimulus. So the signal detection theory assumes that there is no single absolute threshold, and detection depends on person's experience, their expectations, their motivation, and their level of fatigue. Remember what we said, when you're tired, you are not actually as aware as you should be of stuff. So fatigue when you're tired. Your motivation, like you got the radar person here. So is the guy actually motivated to stay and look at that screen and pick up the slightest noticeable difference, when something, a small beep comes across the screen. Expectations, do you expect something to actually occur, and what your experiences are. So signal detection difference theory is a little bit different than all the other theories that we just looked at. So the observer decides whether she hears the tone or not. Based on the signal being present or not, this translates into 4 outcomes, So we look at sensory adaptation. So this diminished sensitivity as a consequence of constant stimulation. So if you put a Band-Aid on your arm, after a while you don't even notice that it's on your arm. It's like you've been wearing your watch or something all day and you don't even remember that you've been wearing it because you don't notice it anymore. So we're looking at vision now. In vision, please make sure you know how light is going to be transferred, how visual images are transferred in the eye. Please know the different parts of the eye. This has been on the AP exam in the past. It could be an essay question, and they would expect you to be able to take the AP reader through the process of vision. Okay, so just make sure you know it. When we look at transduction or phototransduction, this is the conversion of light energy into neural impulses that the brain can understand. So you take the light energy and your brain makes some sense out of it in phototransduction. So stimulus input, the light energy. You guys remember Roy G. Biv. That's the colors of the rainbow. That's the visible light spectrum. So the part of the spectrum that are actually visible to our eyes, here it is, Roy G. Biv. And so typically this is the stuff that we see the most, and then we see them a little bit less, okay? When we look at wavelength, intensity, and saturation, wavelength means hue, color, intensity means brightness, and saturation means purity of color, okay? So wavelength, the hue, that's the color, is the dimension of color determined by the wavelength of the light, and wavelength is the distance from the peak of one wave to the peak of the next. So when you look at the short wavelengths, we look at a high frequency. The bluish color is the high-pitched sound, so short wavelength. The long wavelength means there's a low frequency. So the reddish colors, the low-pitched sounds. Here, this is Roy G. Biv, long wavelength, shorter wavelength. See these? The Roy part, more than we do the Viv part on the rainbow. And it's because of the length of the waves. So intensity, the amount of energy in a wave determined by the amplitude, is related to the perceived brightness. So just intensity, just know it means brightness, guys. So when we look here, the blue color with varying levels of intensity. So the brightness, okay, very, very dark here, keep going, bright, 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 very intense, and then wash out. Purity, saturation. So saturation, what we're talking about is the purity of the color. So green, as you start adding in other colors, it gets less saturated. Same thing with this red. Add in less light or more light, and you get a difference in your colors. So color solid represents all three characteristics of the light stimulus on this model. So here we are. We look at high lightness, low lightness. We look at hue, and we look at saturation. So here is the eye. Please know the different parts of the eye, and remember the little chart that I gave you, guys, to help you out with the eye and how the process of vision goes through. So here's the image. Okay, we have the iris, the pupil, the lens, the cornea, the blind spot, the retina, the fovea, the optic nerve. So the light is going to enter, Okay, and it enters the eye. Okay. And when it enters the eye, it's going to be transduced into an image for our brain to actually perceive. So the cornea is the transparent tissue where light enters the eye. The iris is the muscle that expands and contracts to change the size of the pupil. And remember, your pupil contracts because it needs to let in light. So when it's dark, our pupils get really large so that we can absorb more light. It's why when you turn off your bedroom light, you kind of can't see for a second, and then your pupils get larger to adjust to the light that's in the room. The lens, it focuses the light rays on the retina, and the retina, it contains sensory receptors that process visual information and send it to the brain along that optic nerve. So the lens, the transparent structure behind the pupil that changes shape to focus images on the retina, and then accommodation is going to occur. So accommodation is the process by which the eye's lens changes shape to help focus near or far objects on the retina. So accommodation can be why some people have our vision problems. So we'll look at nearsighted and farsighted. If you're nearsighted, it means you can see things up close. You're nearsighted. And if you're farsighted, it means that you can see things better farther away, and things become a little bit foggy when they get closer. When we look at the retina, the light-sensitive inner surface of the eye, it contains receptor rods and cones in addition to layers of other neurons. So we've got bipolar, the ganglion cells that process visual information. So you have ganglion cells and bipolar cells here for your eye to actually help transmit this image. So the optic nerve is going to carry neural impulses from the eye to the brain, and we have a blind spot on our eye. And remember, all you have to do is take your two fingers, put them about an inch apart, and bring them close to your eye, and eventually you're not going to have two fingers, you're going to have that finger sausage. And that's how you find your blind spot. Everybody has a blind spot. It's just like in your car. There's one part on your car, when you're backing up, you're not going to be able to see, and that's your blind spot. It's the same thing with your eye. So the blind spot is the point where the optic nerve leaves the eye because there are no receptor cells located there. So that creates the blind spot. The fovea is the central focus point, or the central point in the retina around which the eye's cones cluster. Remember, cones are for color. CC, cones, color. So we're going to switch this. So we're going to go to the photoreceptors. And so photoreceptors are in our human eye. We look at bipolar and the ganglion cell. Bipolar cells receive messages from photoreceptors and they transmit them to the ganglion cells, and this is for our optic nerve. So visual information processing, optic nerves are connected to the thalamus in the middle of the brain, and the thalamus connects to the visual cortex. So here's what your brain looks like in relation to your actual eyes. So here's your eyes, and here's how the brain gets all this information thanks to the optic nerve tract, sending it to the visual cortex at the back of our brain. So the ganglion and the thematic cells, so retinal ganglion cells and the thematic neurons, they break down visual stimuli into smaller components and have receptive fields with center surround organization. So feature detection, these are nerve cells in the visual cortex respond to specific things like edges, angles, movement. So feature detection, you can tell a table has edges, you can tell that your computer has edges and has angles and movement in it. Shape detection, specific combinations of temporal lobe activity occur as people look at shoes, faces, chairs, and houses. And so what we're looking at here with shape detection is that your brain has different parts. So when we're looking at the houses and the chairs is right here, faces, chairs, houses. So perception in the brain, our perception is a combination of sensory bottom-up and cognitive top-down processes. So if you're looking at bottom-up, it's sensory. If you're looking at top-down, it's cognitive. And visual information processing, our brain can do parallel processing. We can simultaneously process different things. So we can get color, depth, form, and movement all at the same time because our brain allows simultaneous process, this parallel processing.